So we're going to move on now, only a few minutes late, into the next panel, which is, we've heard mostly about oncology so far. And a year ago, when we wrote the pipelines paper, we literally got the question pretty consistently of, does anyone other than oncology think about doing platform trials? So now you're going to hear about the explosion of new activity that's happening beyond oncology in doing platform trials. And we're thrilled to have Susan Schaeffer here from BioCentury to lead that. Thank you so much.
Thank everyone for joining us for this panel discussion of platforms beyond oncology. My name is Susan Schaeffer. I'm the editor of BioCentury. For those of you who don't know BioCentury, we offer a variety of publications and data products for and about the biopharma industry. Our coverage space is very broad. We are disease agnostic. We cover them all. We cover all of the aspects of concern to senior managers and investors in biopharma companies, ranging from early stage discovery through clinical development, regulation, corporate strategy, finance, politics, and policy. You name it, we cover it. But the meat of our coverage is and always has been the development of pharmaceutical products. And a big thrust of our coverage over the last several years has been innovations in clinical development, including platform trials and other innovations that can not only bring efficiency to the enterprise of developing drugs, but also can produce better information for a wider range of stakeholders. So I'm delighted to be here today and to be with a panel of people who are all working on platform trials in one stage or another. We've heard a lot already today about the exciting progress in oncology platform trials that simultaneously test multiple therapies. These trials are not only producing impressive operational efficiencies compared with the traditional one drug, one trial model, but they're also enabling rapid learning about compounds, about biomarkers, about subpopulations that can be applied as these studies progress, so a true network effect, as we were learning about at lunch. Now, drug sponsors and academics and government agencies are seeking to undertake platform trials in other disease areas where the traditional approaches aren't working very well. These efforts are learning from and building on the work that's been done on oncology platforms, but they're also identifying and solving new challenges that are specific to the disease settings in which they're working. The members of our panel are all either running, getting ready to launch, or planning platforms in a variety of disease settings. I'll introduce them briefly, and then we'll start asking our panelists about why platform studies are the way they've chosen to solve problems or speed progress in their different disease areas. To my left, your right, is Marina Kozak. Marina is a project officer at BARDA, the Biomedical Advanced Research and Development Authority within HHS. And BARDA has proposed a clinical studies network and a platform trial to test candidates for drug-resistant infections. We have Carole Mitnick from Harvard University, who is principal investigator of NTB, an ongoing adaptive platform study that is testing new all-oral regimens to treat multi-drug resistant tuberculosis. We have Rick Strauss from Johnson & Johnson, who is finalizing a protocol for a platform trial that will test multiple internal candidates to treat Crohn's disease. And Luke, we've all already met in the last session from J&J, but in this panel, he is wearing his hat as co-lead of IMI EPAD, which is the European Prevention of Alzheimer's Dementia Consortium. EPAD is running a longitudinal cohort study now and will begin a platform trial in Alzheimer's disease next year. So let's begin with a little description of the platforms that you're each working on and what you're hoping to accomplish with them. Carole, after something like 50 years, we finally have 2 new drugs available to treat tuberculosis. But even with these new candidates, the complexity of the treatment regimens, the length of time they take, the toxicity leave a lot to be desired. And these regimens are very difficult to be used in the countries where the infections are greatest and where there's the most need. What does NTB hope to accomplish, and how does a platform trial help get you there?
Thanks, Susan. So it has been really interesting to me, the discussion today, so thank you. I've learned a lot from people in this room and on paper and now in person as well. And I guess what we're trying to do, one could call a Robin Hood approach. We're not really stealing from the rich, but we are trying to appropriate innovation that's been used for a disease where there is a large burden in rich countries and apply it to a disease that disproportionately affects poor people and disproportionately affects people living in poor countries. So for those who thought TB was a disease of the past, there are still about 10.5 million new cases of tuberculosis annually, about 1.5 million deaths every year. And this effort, the NTB project, is, I think, innovative in a number of ways. So it is the brainchild of a number of non-governmental organizations and academic institutions that typically, the non-governmental organizations typically focus on service, on delivery of direct care to patients living with multi-drug resistant tuberculosis, that is forms of TB that can't be cured easily by the standard 6-month regimen and instead require a 24-month multi-drug, highly toxic regimen that basically comprises medicines that were discarded in the middle of the last century because of their toxicity profiles. And it includes a daily injection for up to 8 to 12 months. So the bar seemed low. And what this consortium of Doctors Without Borders, the non-governmental organization Partners in Health in Boston, and Interactive Research and Development in Karachi set out to do was to really try to transform the landscape for treatment of drug-resistant TB through a project for which we received funding from the UN-affiliated agency UNITAID. And that was to increase access to these 2 new anti-TB drugs, bedaquiline and delamanid, through routine care initially. So according to what the current guidelines are, the recommendation of the current guidelines from the World Health Organization, so within these 24-month regimens, adding 1 or more of the new drugs and then carrying out an observational study to try to supplement the existing evidence about their use that at that point had just come from phase 2 trials. Both drugs received initial indications based on phase 2 trials, and they were conditional approvals both by the EMA and the FDA. And then the second part of the project is to really, as I said, transform the way treatment is delivered. So I mentioned the 24-month regimen. What we're looking at is are 9-month all-oral regimens in a Bayesian adaptive trial run in a subset of the same sites where the observational study is ongoing. It's 5 experimental regimens, and the hope is that there would be additional compounds to integrate into this platform over time, but there is very little activity in the TB drug development field. There may be some new compounds, but at this point, the platform is more about the observational study, the clinical trial. We are also awaiting a funding decision on a second trial that would use the same screening practice as for the first trial, but for a different subset of resistant TB patients. There are also other efforts underway to use that same platform to bring in new innovations. TB is a very notoriously slow field in terms of uptake of innovation. And so there are some new diagnostics where the uptake has been really limited, and this platform can also be used for that, as well as for really major public health interventions that would be much more comprehensive and much more patient-centered, focusing on prevention as well as treatment of active disease.
I'm going to jump down to Luke at the end of our table here. Luke, after a series of well-known clinical disappointments in Alzheimer's disease, I think it's become very clear that we would be better off intervening much earlier, which raises some challenges in drug development because of the complexity and length of the potential path of progression. How do you identify patients? How long do you need to follow them to get an answer? How is EPAD hoping to address those challenges in this very early population? And why is a platform trial useful to you in trying to do so?
Yeah, thanks for that question. So what you just said is actually the reason why this needed to be a public-private partnership. Nobody would be able to do this on their own, because what we were thinking of doing was picking up asymptomatic individuals out of society and trying to then track them over time, characterize them, see if they have amyloid in the brain. As most of you know, in the trials that failed, up to a third actually didn't have amyloid. So try to find a clinical signal then. So the way then EPAD is trying to address this, and again, we needed academic stakeholders, patient advocacy, and industry coming together, was hooking up with existing registries that are already tracking people over time, and build a relationship with that parent cohort, as we call it, so that we can identify people that, according to certain algorithms, and this is part of the learning, the algorithm can shift over time. By the way, this is going to be my a bit of urgency I have. I hope Austin has a good business continuity plan, because I think the berries are in every single one of our adaptive trials. So hopefully nothing goes wrong with berry consulting. We build an evolving disease model, and we can adapt, our risk scores. Anyway, so we can pick people up from these registries and then enroll them in the longitudinal cohort study that we have running, which aims to track people for at least 6 months, that's the minimum, but up to 3 years, and see how they evolve. They get characterized for risk status and amyloid status. And then, and that's the step that will start next year, for which we're excited, out of that register, we are able to directly recruit into an adaptive trial. So that should massively reduce our screen failure rates. Our current end is, for those who do not know, today, 9 out of 10 people screen fail. We hope to bring that down to 2 out of 10. There's always other reasons. And start running an adaptive trial, as we've had many examples. It will follow the Berry doctrine, so time machine and all that is included. We ultimately, our hope is to show clinical proof of concept. That's the end, that's the exit point for the adaptive trial, because we've not been able to do that in very large experiments, so we want to avoid that cost by doing this in a proof of concept setting. So we can adapt on intermediary phenotypes, as Simon Lufson likes to call it, which is biomarkers we've identified. So that's kind of the setup. We currently had 402 subjects, I think, in the longitudinal cohort study. And the first compound will start second half of next year, which is from our friends of Boehringer Ingelheim. And I think this last point allows me to make 2 points. One, for those who do not know, IMI is actually half funded by the European Commission and half funded by FPA. And that is why the IMI consortia differ from most consortia we've heard about today, which is that it's actually co-led by industry and academia. And I think that leads to a very good tension in terms of finding, okay, what are our needs? what's the scientific community's position to really come together and get to the best outcome. The second thing is platform trials require, for those that invest in that, a certain form of altruism. So Janssen has been driving as the co-lead this EPAD project, yet the first compound in is not ours. And that requires some education within our companies as to how acceptable that is. But we will get there.
Very good. Marina, there are many challenges in testing candidates for drug-resistant infections, but among them, we know that they're relatively rare. It can make it difficult to find patients. And then when you do find them, there's a need to act quickly to treat them. You can't sort of wait a period of weeks to enroll them in a center that's participating. Can the clinical trials network and the platform trial that BARDA has contemplated help solve those issues?
Yeah, so I think, so first of all, thank you for having me here on this panel. I think the issue of rare patients is an issue that's shared among a number of different diseases. So it's not specific to bacterial infections per se. The pace of the infection is a bigger issue. You want to get patients before they're significantly ill, but at the same time, by the time you identify a resistant infection, they are quite progressed. And so having a platform could potentially extend some of the sites into the community where these infections are actually happening. On top of that, another complication is that because of the economics of antibiotic development, a lot of the large pharmaceutical companies have actually exited the development space. And so what's left is some of the smaller and medium biotech companies with very limited resources. And so by providing this a shared platform of infrastructure where companies can share resources, share patients, information about patients, and utilize patients in a much more efficient way could potentially provide a huge benefit. And so that was the idea for scoping the platform trial for BARDA. We're very much still in the scoping phase. There's no protocol developed yet, and we're gathering information.
Right, and I know that there have been some challenges that you've encountered, which we'll get to in a little bit in the sadder part of our panel discussion, where we talk about the difficulties, but we're also going to talk about how those can be worked through and what the next steps are. So we'll come back to that. Now, Rick, J&J has run many trials in Crohn's disease in the traditional way, one drug one at a time, and has approved products to show for it. So why is it that now you're looking at taking a platform approach with the compounds that are in your own portfolio? And if you will, is there a vision to expand that out beyond J&J?
Yeah. So an important question. So I think there's sort of three key reasons. One is just traditional drug development, phase 1 followed by phase 2A, proof of concept, followed by phase 2B, dose ranging, followed by phase 3 just takes too long. And if you look at that, phase 1 and phase 3 are standardly choreographed. You can't do too much in there. It's phase 2 that's generally the bottleneck. And for instance, ustekinumab or Stelara, which was approved for Crohn's disease last year, spent about 8 to 9 years in phase 2 testing. So we have to be more efficient at that. Second, for future compounds, we're looking for superior efficacy. We already do now have a number of moderately effective compounds there. In the future, planning for something 5 to 10 years from now, you need something with superior efficacy, and none of your preclinical models can predict that. No matter how good your animal models of colitis are, they do not have Crohn's disease, and you just are guessing and extrapolating to human disease. And if you take that approach, you do have to kiss a lot of frogs to find a prince. You have to cycle a number of compounds through to understand which are the most effective. And I think the third key issue is the trial design for Crohn's disease has really crystallized in the last 3 to 4 years. It doesn't matter if you're Janssen or AbbVie or Takeda or Pfizer. All of the trials will have symptomatic endpoints, endoscopic endpoints, somewhere between 8 to 16 weeks for proof of concept. So what we found ourselves doing is essentially reinventing the same trial over and over again, trying to put in a couple bells and whistles, but losing a tremendous amount of time, essentially just sort of doing the same trials over and over again. So I think the key question is not why we're doing this now, but why we didn't do this 2-3 years ago. It seems apparent now, and I think the one concern is that would have been nice to have the first couple compounds through the platform, because it takes about probably 3-4, not till you're at your third or fourth compound to really get the value out of this. I think long term we view this as a cross-company collaboration, would be a benefit for everyone, but we thought we just have to start and get the learnings and the infrastructure developed sooner than later.
And you're in the enviable position of having multiple candidates in-house that you can run through this platform.
You have multiple candidates in-house, but they all do, you do cycle through them quicker than you would expect when you get toxicology results or other drug-drug interactions. So you do need to have a very robust pipeline to do this internally, or you have to have plans to eventually spin this out to either business development partners, which is what we're doing now. Is one of our pitches to our partners would be that we have this platform that we can plug your drug into, and that we have sites already up and going. And now that's the plan. We're planning to start later by the end of middle of next year.
Now, I think that we've all learned a lot from the oncology platform trials that come before, certainly from I-SPY. We've all learned a ton. Are there specific things that you've looked at these oncology platform trials that are ongoing, some of which are producing results, where you've identified key lessons or enablers that you're applying to your platforms in these different diseases? Luke, you want to start?
Well, I think that, as we heard, I mean, the key is the accelerated learning you get from more and more molecules coming in, and how you're actually honing in on, okay, what's the background rate against which, so that ultimately you might even drop control groups. I mean, just imagine. If regulators would accept that, that would be fantastic. But it requires a very robust data set, and by going through this with multiple compounds, we got there. I just want to make a comment also on regulatory interaction of consortia in diseases with high needs. I must say the regulators, whether it's in Europe or in the US, have been very, very positive and constructive. In EPAD, for example, where you had to have scientific advice on the first hand, we had a very robust discussion with CHMP that you would never be able to have as a single company. And by virtue of the huge unmet needs and how we construct things, we got pre-agreement, let's say, that the longitudinal cohort study data could be our run-in data for the randomized experiment. Try to get that done on your own. And then in the execution phase, the clinical trial facilitation group helped us out with how do we now, who is the sponsor, how do we manage these CTAs and all that. I think I want to acknowledge that regulators really have a positive role here.
And apart from everybody working with Berry Consulting, other lessons from the platforms that have gone before?
Yeah, I think having the publications out and having, even last month or 2 months ago, New England Journal of Medicine article, extolling platform trials, this type of conference, getting it on people's radar screen really sets the groundwork for expanding this to other indications.
I would add, we are actually not working with Berry Consulting, I will confess. But I think one of the things that for me was a little bit eye-opening and was really helpful is I had made the assumption that these platform trials were only possible in cancer because there were excellent biomarkers for all cancers. And turns out that's not so true. And so that was reassuring because we're working in an area where we have at best weakly validated biomarker, but through the Bayesian adaptive randomization are able to have the priors updated with information that correlates the interim response to the later response or provides more information about the correlation between those as we engage in the trial. So.
Well, that's a great segue into some of the key differences between the diseases that you're all working in and some of the platform trials that are ongoing in various cancers. I think Laura Esserman said earlier that, why are we running several hundred patients through a study to find out that the compound doesn't work? We can do that with 50 patients in I-SPY. Laura has, biomarkers that can be used to assign patients to different arms, and then also an early endpoint in PCR that can provide for a quick readout, and an endpoint that's been correlated with a longer-term outcome of interest. Are any of you going to be able to make decision-making based on 50 patients in an arm? Do you have the endpoints? Do you have the biomarkers? And if not, how are you adapting?
Yeah. Oh, sorry. Oh. You are in a much more comfortable position than me.
I think in Crohn's disease, we could declare futility with 50 patients. You need probably more patients than that to really know where your success is and whether you're going to be superior. And it does raise some questions about with adaptive randomization of where you should be channeling your patients. If you have two compounds in here, and compound 1 has some efficacy, when compound 2 comes along, are you better off channeling to that first compound, or do you want to still channel to that newer compound? And if it's ineffective, part of what we want to do is get ineffective compounds out of the system, and it may actually be speedier to not adapt to that first compound because they just need more patients. But it's a tricky question to understand that difference with our disease versus oncology, where you do have the clearer biomarkers.
Right. And do you have sort of early and late endpoints, as Laura described, in Crohn's?
All of our endpoints are probably about 12 weeks, so they're not these long-term 1 year or 2 year endpoints. Okay. So we can get to our symptomatic endpoints usually within 4 to 8 weeks, our endoscopic endpoints within about 12 weeks or so.
Got it. Luke?
Well, the lift in Alzheimer's is about an order of magnitude bigger. And the reason for that is twofold. One, we do not have validated surrogate markers. Two, because we're going early pre-symptomatic, and we have agreed that the endpoint has to be a clinical endpoint, we are following them for about 2 to 3 years. So we have to account for dropouts, etc. So the average size of a assignment for a molecule is 500. Which is still very large, but it's unfortunately the way the situation we find ourselves in, which is why I'm going to stand on a soapbox now. I'm a bit of a data communist. We need to share, and this is talking to our company colleagues, much more our active data so that we know what we have. On the one hand, do we have a racehorse or a donkey? Because there's no point in pouring resources in things that meet the threshold just but are outstripped by their counterparts. A lovely conversation I have with lawyers in the company, I will tell you. And secondly, because we don't have the biomarkers, if we share our active data and the same biomarker tracks in two, we need to know that as fast as possible. Because that's, and Bob can correct me maybe, but that's a requirement for the validation across multiple mechanisms. So we have all to gain from being as open as possible with these platform trials. So I'll step down now.
Carole?
Whether we like it or not, we are always doing cross-study comparisons, and
Yes.
we're limited by different design, different countries, different sites. So at least now you're able to compare your drug within a common framework,
Right.
and that's better to know earlier than later.
Right, which is a shift in the way that many companies have thought, and a fairly recent shift. I mean, we didn't even start seeing kind of head-to-head phase 2 comparisons against active comparators until maybe 7 or 8 years ago. Now it's hard to find a company that would even think of moving beyond that point without having some comparison to ensure a differentiated profile in the marketplace. And it's true, physicians, investors love to make cross-trial comparisons. Isn't it better off if we're doing it on the basis of data that are actually more similar than they are different?
Absolutely.
Yeah. Carole, in TB, this early versus late endpoint is a particular challenge. Why don't you just describe that and how you're accommodating that, or what you're adding into your study to sort of maybe identify some endpoints that could be measured earlier in the future?
So just thinking about the time scale that Rick just mentioned and for their early response. So the earliest response that has any correlation with final endpoint is at an 8 week after treatment initiation, and then it takes another 6 weeks to confirm that the bug hasn't grown. TB is a slow-growing bug. It's a slow-moving field. And so we cannot we do not have the ability to acquire information on, say, 50 patients per arm, mostly because we don't have this particularly well-validated marker. We have, however, one of the advantages to the Bayesian adaptive design for our question is the efficiency, and that it's about 20 to 25% more efficient in terms of sample size than a fixed randomization design would be for our attempt to identify multiple potentially non-inferior regimens. So in the course of the trial, we, as I mentioned, will be able to gather information about the correlation between the 8-week endpoint and the 39 and 73-week endpoints, and then use that to update the prior, and ideally combine that information also with other trials that are doing similar work. This endpoint has been a little bit better validated in drug-susceptible TB, but not so much within drug-resistant TB. So our 750 participants will contribute fairly substantially to the foundation of being able to develop.
And is this the sputum test?
Yeah.
Okay.
So everything is sputum based, and the bug has to be cultured.
Right. But I think, if I recall correctly, you're in the fortunate position of having at least one pharmaceutical company that is investing in helping to validate that biomarker with partners and plans to take it through a formal qualification.
Yeah. So one of the developers of one of the new anti-TB drugs, Otsuka, is also working on validating a biomarker, and it's not clear that really for drug development is their objective. There are some challenges. Not all industry has what would be called communist attitudes towards their intellectual property or their data.
Viewpoint.
Yes, I understand. So being a consortium of non-governmental organizations that's really committed to open access, that's one of the issues that we have a little challenge negotiating with industry is how the information would be used if we were to provide samples to validate that endpoint and also that marker, where the proceeds would go. That's an issue for our consortium.
Mm-hmm. So I do want to spend some amount of time, not to dwell on it, but discussing some of the challenges or barriers or surprises that you've each encountered as you set out to design and launch platform trials. And I'll start with one that has already received a certain amount of discussion today, which is how do you finance these things? Because I think when we talk about these platform trials, more conceptually, we tend to focus on the efficiencies, that they're faster and cheaper. But in some ways, it strikes me that what is really happening here is similar to what we see with the development of curative therapies, where what you're doing is it may be cost effective, but you're shifting the cost up front. In a way that we don't really know how to fund. And Marina, I want to start with you because I know that BARDA sent out a request for information on costs, and the findings were unexpected.
Yeah. So that's right. So BARDA put out a request for information for a 10-year period of performance, and the interest was, what is the value to industry, and would industry be willing to participate in an effort like this? And so the ask was to create a clinical trial network for approximately 3 antibiotics on a rolling basis annually, and this would be per indication, and would cover 5 of the most common indications. And so what the average costs that we received back, and so just, the answers are coming from industry. And so we didn't do an internal assessment of what those costs would be. But so the total cost for a 10-year clinical trial to start up and maintain a clinical trial network for each indication would be approximately $100 million. And so that would include site selection, screening, clinical monitoring, network management, training, some of their clinical research infrastructure. And so that was a very surprising and alarming amount. And so for us, our funding comes from Congress, and so there would need to be a significant lift to get that pushed and included into our budget. And so at this point, we're still considering how best to go about this, but the costs are incredibly prohibitive.
And as you mentioned, many of the companies that are currently, I say many, there aren't that many, the handful of companies that are developing novel antibiotics that might be applicable to these drug-resistant infections, they tend to be the little guys. There are a couple of pharmas that are either still working in this space or have recently re-entered the space, but mostly they're smaller companies, so you don't necessarily have the potential to tap into industry funding.
That's right.
Yeah,
that's right. Yeah, limited resources. And for perspective, there are currently 37 antibiotics in phase 2, phase 3 development, compared to in oncology. That's a huge difference.
And this is another, and you guys are just feeding the segues. That's great. That is one of the issues in some of these diseases, is having enough candidates in the pipeline to feed a platform trial, make it efficient. And Luke, Alzheimer's disease, we've lost a lot of candidates over the last couple of years.
Yes, unfortunately, but the pipeline early on is growing again. So, and I hope we will actually be ready in time to not be a bottleneck. I do want to come back to cost. You say 100 million. That sounds like a lot. But if you talk about having a standing infrastructure that stands for 10 years, and you would say maybe 4, 5, 6 come out of that, that's an easy repay. I mean, it's a big number, I don't misunderstand me, but in the scheme of how development works today, it's EPAD's project is 60 million euros over 5 years. Right, so I mean, it's in the ballpark. That's what it will take, and that's why I'm saying you need to go in an area of high unmet need, where there is an emerging pipeline, and where you can bring stakeholders together that are willing to share the risk. That's, I think, what you need. And in Alzheimer's, clearly that was there. Hopefully for this infection, TB, of course, we also put a lot of work in that one. But yeah.
We don't find the cost, at least what we're projecting for, say, 3 separate trials versus 3 compounds within a platform, seem to equal out even up front. I mean, we do have to redesign our systems to allow for crosstalk between compounds, but that's internal.
Right.
Cost. I think the real cost will come up if you don't have, if you're keeping the trial open without having a compound feeding into it. That I think we've been told or warned is that if you have spaces between compounds is where the concept can be challenged or fall apart or the cost go up. But if you have enough drugs to put in there, the cost should work out to be about the same.
I'm going to open this up for questions shortly, but one more for the panel here before we do. If you were starting from scratch now, are there things that you would do differently, or alternatively, what do you know now, given the amount of work you've already put into these trials, that you wish you'd known at the beginning? Either question that you like, and we'll just go down the line, starting with Marina.
Well, we're still in the scoping phase, so I would say for us, given the number of products currently in development, I think perhaps platform is potentially not the best way to go, just because there just wouldn't be enough drugs to sustain the network. But I think potentially there's an opportunity to start incrementally, as Janet Woodcock earlier alluded to, and incorporate small adaptive elements into existing trials, and start small and build up. And hopefully in the future, as the pipeline expands, there might be an opportunity to make this bigger.
Is there any interest at this stage from companies that are getting closer to the right phase with their candidates to do sort of a scaled down version, so not maybe the full network, but to be able to get some efficiencies out of screening and control arms? Or is this kind of an all or nothing proposition?
No, I don't think so. I think there is definitely interest. There's absolutely interest from the FDA in companies pursuing this. We've had a lot of positive feedback. There's some interest in industry, and I think the Berry Group actually is trying to tackle some of this. The Antibiotic Resistance Leadership Group has also been heavily involved in incorporating some of these adaptive elements into trials. So there's interest, and it's moving forward, even if it's not through a BARDA network specifically.
Carole, something you wish that you'd known when you started that you know now, or something that you would do differently if you were starting from scratch?
I think the biggest issue is the issue we were just talking about. For us, it's the funding. So we've been funded by an entity that really, made a strategic investment in building the infrastructure for this platform, but their mandate is to catalyze market change, and they're expecting results in 3 to 4 years. And I think we probably would have looked for funding from multiple agencies had it been as clear as it is now that was sort of their expectation. And so we risk having built up all of this platform infrastructure in 6 countries on 4 continents, and having limited use for it when these initial 2 efforts finish.
Rick?
I think the biggest thing that we've learned is how much education is needed outside of the oncology area to get a site. The average Crohn's disease site recruits 1 patient per year, so you need 100 sites for 100 patients, and you have to approach. Normally we'll send out questionnaires from our clinical operations to to 500, 600 sites, are you interested in our trial of this compound? Now, if you do that with we're starting a platform trial, we can't tell you which compounds yet, but we could tell you one, and are you interested? They don't know what you're talking about, and it takes a lot of, and same as ethics committees, I think outside of oncology, they're hearing about this, but trying to understand the consent process and how you're going to consent to multiple compounds and take away patient choice, where they would normally choose between which compound they think they'd rather enroll in. I think it just takes a lot of time and education and efforts to reach out even globally, because we do global programs about the platform trial. And again, I think spreading this out from oncology and learning from oncology has set the framework, but it is still an untapped field in IBD or Crohn's disease, so it does take a lot of that effort.
I agree with everything that's said. In addition, education of tech transfer offices of universities. We have spent months negotiating about things that shouldn't be negotiated because it's partly public funding. So anyway, that was eye-opening for us.
Luke, tell them how many members of the consortium there are that you're working with.
37 now. Merck is joining us as of next year. Yeah. So I'm happy with that. So
that's a lot of negotiating.
Yeah, but within companies, I mean, it was mentioned before, there was no problem at all. We got that project agreement. It's then going into the academics, where the distance between the enthusiastic academic that signed up for the consortium and the decision maker on whether or not to accept the IP language is miles. And we lost months. So that's.
How did you ultimately address that? Were you able
Sometimes the centers had to drop out. We lost an important genetics component because we couldn't get agreement. It was very painful. I want to end with, I think there's going to be one key enabler for us to be ultimately successful, and that is whoever consents a subject research participant for anything, let's make sure that that informed consent contains a recontact clause. Because that's the other big thing we had to overcome. How can we get back in touch with people that at one point were saying we're participating in research, and we can't even, by rule, recontact them if it's different research. So, and we heard from advocacy groups is that, hey, we're here, we want to do it. So one way around is actually the Critical Path Institute that is leading initiative to build a universal language. And I think we, as all participants in this endeavor, should include that, so we don't see that hurdle.
Do we have any questions from the audience for our panel? We have a few minutes.
This is a very interesting, Bob O'Neill, FDA, very interesting conversation. I attended, the reason why I'm bringing this up is what lessons learned for the rare disease, orphan disease population, because essentially this conversation is all about the use of a randomized design and not the use of registries for historical control data and all that stuff. And I attended a NIH NCATS meeting about 5 months ago on data standards for registries, and all the patient activists were interested in what data should I collect so it will be useful to someone. And they're struggling for what is it going to be useful for. A lot of folks would like it to be useful for market entry and maybe use as controls or whatever. Do you guys have any sort of messages for how to bring the NORD crowd on, that's the rare disease crowd, in terms of the value right away to think of registries as over here to set up your platform trial at the end of the day, because that's what you want to do. You don't want to use a historical control. Use your registries as a historical control for a single-arm trial. Oncology's been there, done that, and want to move on. So how does sort of that message get to the orphan disease crowd? That's.
Well, I can't talk to the orphan disease crowd, but I can talk that in our AD space, We, of course, have a lot of legacy registries we're trying to capitalize on when we have the hurdle. But in the context of the UK, with UK Biobank, for those who don't know, it's a 500,000 people register that's live now. We're building a registry extract from that will exactly look prospectively at all the same measures we're looking at in the planned adaptive trial, so that we can have some comparison. And as Janssen, we ourselves are running one. It's called Chariot Pro, about exactly doing the same thing, actually running a trial without a drug in a way. Yeah, but it's not orphan drug, orphan indication. I don't have insight on that.
In Crohn's disease, a lot of the registry questions come up with pediatric development and how we can facilitate pediatric drug development, because pediatric ulcerative colitis and Crohn's are orphan diseases. Can't say we have made a lot of progress yet. Yeah.
And I would just add that what we've seen in the field of multi-drug resistant tuberculosis is not so much registries. I mean, much of TB is treated in the public sector, and there is a standard limited amount of information that is collected by those public sector programs on patients, but often clinicians will do retrospective cohort studies on the patients they've treated, and there is now a 17,000 individual patient data meta-analysis that has been assembled painstakingly over the course of many years by some investigators, and that has produced a lot of information sort of in the interim while we're waiting for clinical trials. And there's some discussion now about the appropriateness of that as a comparator for trials. I think that the biggest issue that I've seen is that those are data that were collected during, from 1999 to 2009, and there's a single-arm trial going on in South Africa right now that wants to use that as a comparator, but it's not, it's temporally completely different, and we've seen enormous changes in the performance of the control arm in the most recent trials that reported, going from about 50% to about 80% response in the control arm. So it seems like it would be really risky to use that as an historical control.
I would hope the Nord crowd would recognize that message for the orphan disease. It's not an easy thing to decide upon, but the value to the data is what it is.
Robin Lim from Health Canada, Health Products and Food Branch. Thanks very much. It's a very informative meeting so far. I have a question that relates a bit to an earlier question, I think, that Bob from Georgetown had asked about building in that sort of voice for creativity that might not have been thought about. And I'm just wondering about upstream in designing these platform trials. How collaborative is that process to think about what all the various stakeholders' needs are in what you're actually designing to come out the other end?
Well, there is a limit to the creativity, of course, if you're in a trial. But, for example, additional questions around exploratory biomarkers. So we're banking a lot of things. So we have a sample access committee that looks at what are you proposing? Is it a good use of this restricted resource, and then do the additional experiment. Part of what we're setting up is actually not just a platform trial, it's a site network. So they could always maybe have additional bandwidth that they could do additional stuff, and you still have an efficiency from common training, common standards, and this and that. So we are not, this is never intent to stifle creativity. It's just we have to have certain rigor to get to the main goal as well. But I'm not sure I'm fully answering the question, but there is some possibility for additional biomarker work for sure, and then just utilizing the sites.
And in Crohn's disease, we know some of our endpoints are shifting, that some of our older PROs are not going to be accepted in the future. So we are trying to build in some of these newer PROs, and this way, instead of just testing them as one-offs on individual compounds, we can see how they perform across multiple compounds. Again, having some common biomarker collections, tissue biopsies, we can understand how different compounds affect disease biology. So we are trying to look not just to do proof of concept, but to anticipate some of the questions that'll come up 3 to 5 years down the road.
And maybe along these lines, I mean, is it naive to think that if you've built the infrastructure, if you've standardized the protocols, the data elements, the ways that the data are warehoused and handled, does that not free up some time and energy for more creative pursuits in the future? If you're not recrafting the trial every time, you can maybe be thinking about more interesting questions that will yield more useful answers. And if you have a flexible platform, you can build those in as it progresses, yeah?
Yeah, I would say that seems plausible. We're not at that stage yet, so I can't speak from experience. I guess the other answer I would offer, Robin, is the kind of consolidation that was described, or that I understood from the last session around those two particular indications, that has not really happened with our setup around multi-drug resistant tuberculosis. There are multiple consortia, multiple networks still pursuing clinical research, so there's still lots of room for innovation outside our own, as well as inside, with sub-studies being proposed and and different banking specimens for looking at different biomarkers.
We're here.
Hi, Ed Kozak, payer strategy consultant. So there's several things I heard that will be new to payers. First off, they don't know much about platforms, and you mentioned adaptive randomization and some other things along the way. What steps are you taking to receive input from payers and health technology assessment organizations?
I can talk about it in the IMI context, where this is not all within the same project, but it's in the Alzheimer's strategy, where we have a project called IMI Roadmap, which looks at outcome measures, what's relevant, and NICE is actually a partner of it. So, and they will all feed each other. We're going to launch a concerted support action, which means some extra overlay to coordinate all these projects in Alzheimer's space, which is EPAD, AMIPAD, Roadmap, I'm missing EMIF, ENS, Atheonomy, all in Alzheimer's, and we're going to make sure they all share.
I guess just from my perspective, I think it's very different because, as I mentioned, the payers are largely public sector, so it's meeting with ministries of health and directors of national TB programs to try to explain what this means and how the results from a Bayesian adaptive randomization trial, for example, still can produce frequentist effect estimates that can be used in the same way.
I think we use this data we need to know where to set our goalpost and how high we need to aim, and that just sort of be in the same. I mean, we've heard loud and clear, just being the same as what's out there today is not acceptable. So we have to design our trials for the future and the future payment landscape.
And the trial that you're designing now, this is a sort of a phase 2?
Phase 2
yeah.
program.
So at what point would you typically be talking to payers about the programs in your pipeline?
It's a good question. I think certainly after you need to have your initial data first of where you think the goalpost would be. So after either phase 2A or 2B, while you're ramping up for the phase 3 program.
Since there's so many different multi-diseases, I thought I'd ask about manufacturing and scalability of the manufacturing. Are there any comments around that with respect to batches of drugs, and in particular any of the scale-up issues, which would also be one of the things to think about when you're talking to a payer before you would scale up?
I hope to get a proof of concept first, but
I'm interested in the TB one.
So, and sorry, were you talking about manufacturing within the context of the platform trials, or
Yes,
follow?
because you've got the batches
Yes.
of the product, and they have to
Yes.
have specifications.
Yes, we do. We do have a little bit of a challenge in, at least with the drug that's manufactured by Janssen. Not to call you out at all, because we're very grateful for Janssen's collaboration in this. But Janssen has entered into an agreement with Farm Standard for distribution of the drug in the former Soviet Union countries, and we have 3 of those in our clinical trial, and really want a single source for the trials for all of the sites. So we were able to negotiate that with Janssen for the first of these efforts, and are still in the process of sorting that out for the second set of regimens that we would be testing. So
Just as a general comment of interest, I'm just interested in a platform trial environment where you're testing lots of things. What would be a strategy around scale-up if you were going to do that? Because you probably start with small amounts of product until you want to.
I mean, we're not, and I am sorry, I can't really speak to manufacturing, and we're not starting small. We're starting with 750 patients in one and 2,600 patients in the observational study. So it's, I mean, by trial standards, it's not too small.
And these are all commercially available products.
Yes, that's an important point. These are already commercially available.
Right, and you're looking at novel combinations, but all of the products are available, so presumably they've been scaled up.
There are some manufacturing issues still, because that's part of the objective of the whole process is to scale up use more.
I think if we are getting back to trying to limit the time it takes to go between phases, you do have to do some scaling up at risk. If you have to wait till your proof of concept to start manufacturing your phase 2B supplies, or wait for phase 2B to start manufacturing phase 3, you are going to add in another 10, 12 months between stages. So a lot of that scale up is being done at risk.
Well, I think we're just about out of time, and hopefully we've gotten us a little bit back on schedule today. I want to thank all of our panelists very much for the discussion.
Okay.
Thank you all for listening.
Thank you.